Hi, in this video we're going to be talking about genetics dihybrid crosses. So remember when we've been talking about Punnett squares, we've been talking about chromosomes, and chromosomes are just folded up DNA. Remember that you do get a copy of all of your DNA from dad, a copy of all your DNA from mom. Mom and dad cells, the sperm and egg, are both haploid cells. They come together to make you, and you are diploid. So let's look at these two chromosomes right here that are blue. They are homologous because one came from mom, one came from dad, and they contain the same genes. Remember, genes are areas of DNA that code for a protein. So let's look at them down here. We've got the gene for color, and in this case from one parent you got purple. The other gene is also for color, and it is white. These are homologous because they contain the same genes, but they have different alleles. If you have the same alleles, this would be called homozygous, and if they were both dominant, homozygous dominant. If you had the same alleles and they were both recessive, homozygous recessive. If you have different alleles, which is what we have here, this would be heterozygous. Recall that the word phenotype means how you physically look. So for example, saying the word purple, saying the word white, that's the phenotype. Saying the genotype would say that purple is dominant and it is letter capital A, and then white is recessive, it is letter lowercase a. That would be the genotype. All right, let's revisit the steps of meiosis. So in meiosis, we start with a diploid cell, meaning it has two copies of every chromosome. It has a mom copy and a dad copy. They're homologous. When we go through interphase, we go through G1, we go through S, which is where my DNA is copied, and then we go through G2. Let's focus in on S for a quick second. What does that really look like? So if we look at my homologous chromosomes right here at the beginning, notice that one of them has capital A, capital B, one of them has lowercase a, lowercase b. When I go through replication, oh, that's my alarm. When I go through replication, we copy the chromosomes and make them sister chromatids. So in this case, the green ones, these sister chromatids will be identical to each other, and then the purple ones, the sister chromatids will be identical to each other. So let's look at that. If this one has capital A at the top, its sister chromatid will also have capital A. If it has capital B, its sister chromatid will also have capital B. Same thing for the other one, lowercase, 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 lowercase. So because I've gone through replication, which means I just photocopy it and stick the two together, I get 4 sister chromatids with 2 homologous chromosomes. So remember, the whole thing is called a chromosome. Each half is called a chromatid. All right, so now that we've done S phase, we've copied our DNA, let's jump into prophase 1. Remember that during prophase 1, we do crossing over. So crossing over would take portions of the chromatids of different homologous chromosomes, or sorry, not different one, but of homologous chromosomes, and they would swap them. So again, sister chromatids of homologous chromosomes are swapping. Once we do crossing over, then we're going to go through the rest of our phases. At the end of telophase 1, I'm going to pull my homologous chromosomes apart. At the end of telophase 2, I'm going to pull my sister chromatids apart. So let's see. So here is the end of telophase 1. I've pulled my homologous chromosomes apart, and notice because of crossing over, they are unique. And then at the end of telophase 2 right here, I've pulled apart my sister chromatids. I end up with 4 haploid gametes. And in this case, all of my gametes are unique because we did crossing over. We want our children to be unique. We don't want our children to look identical. So this is why we do crossing over, and this is why we make 4 haploid gametes. So when we've talked about Punnett squares before, we've only done 4 boxes. So for example, let's say these are all eggs, so the mother. We're going to place them here. Why do I only have 2 boxes if she has 4 different eggs? Well, if I notice, remember we've done our Punnett squares, we've only used one letter at a time, so let's use the letters A. If I notice, she has capital A on 2 of her eggs, she has lowercase a on the other 2. So these 2 are repetitive, right? So instead of making 4 boxes on this side for mom, I'm only going to make 2 boxes because 2 of her eggs will have capital, 2 of her eggs will have lowercase. And then whatever the father is, I would have to find and put those at the top. But the real reason I'm bringing this up is because I want you to understand that even though she makes 4 eggs, we only write 2 on the side because they are repetitive to each other. Now, this is called monohybrid crosses because mono means 1, and we are only looking at 1 trait. We're only using 1 letter, like the letter A. We didn't do A's and B's at the same time. We're just doing A's. But what if I do want to do A's and B's? So now we're going to be looking at dihybrid crosses. Di meaning two traits or two genes. So now we are going to look at A and B at the same time. So let's say again that these are all eggs. So since I'm looking at A and B, I'm going to write down the different combinations of A and B that mom put in her eggs. So we notice that mom put a capital A and a capital B in the first egg. She put a capital A and a lowercase in the second, little with capital, and then little with little. So now I have to have 4 different boxes on the side of my Punnett square because she does have 4 totally different gametes to put into her eggs. And then again, I would also do the same thing for the sperm of dad. Now, let's talk about something weird. Right here, we put a little a first and a capital B. Why didn't we do capital B first and then little a? The reason is we've been doing A first the whole time, so I can't swap up the order even though the A is lowercase. It's okay. If we've been using A first the whole time, please continue to put A first. All right, so let's actually work a problem. So here are the steps. Pause, write these down in your notes. They are almost identical to the ones that we've been doing before, but there's something a little bit different called FOIL or 2 Punnett squares. So do write these down and then let's go ahead and solve a problem. So round is dominant to square and yellow is dominant to green. If a round green and a round yellow pea plant are crossed, what percentage of their offspring would be round and yellow? Well, first step, obviously, we have to underline our question. So what percentage of their offspring would be round and yellow? We need to box our traits. So you could be round, you could be square. You could also be yellow and green. So now I have 4 different traits that we're talking about. We have 4 alleles. 2 genes, but 4 alleles. So I'm talking about color and I'm talking about shape. Step 3 is to obviously underline our parents. So our parents would be round green, and it already tells me their genotype. Ooh, red was bad color for that. Let's choose a light color. Alrighty, so round and green, and then my other parent is going to be round and yellow. And again, they've already told me the genotype, so I just have to roll with it. Alright, so now that I have annotated my question, I can go ahead and write down what my letters stand for. So trait 1 is for shape. If you capital R, you're round. 2 lowercase, you're square. Trait 2 is for color. If you are capital Y, you're yellow. If you are 2 lowercase, you will be green. Now I need to write down my cross. So I'm going to say parent 1 is this one right here, the round green. It is capital R with little y's and a little r. I want to get all the combinations that this parent will put into, let's say it's a female, into her eggs. So capital R can go with little y. Capital R could also go with this little y. Can anything else go with that first capital Y? Or with that first capital R? Nope. Go to the second R. So little r, little y, and little r, little y. So that means in the egg we'll have a big R with a little, we'll have a big R with a little, and then we'll have a little with a little, little with a little. Let's go to our second parent. It was big R with big R, big Y with little y. First R can go with capital Y. Helps if I write it correctly. It could also go with lowercase y. Nothing else can go with that first R, so I move on to the second one. Big R, big Y, big R, little y. Let's go ahead and fill out our Punnett square with all this information that we have just gone through. So again, we know that it is big R with little y for this parent right here. It is also big R with the second little y. We've got little with little and little with little. Let's go to the second parent. Big R, big Y. And I'm just copying this down from the last slide, whatever we wrote down. Big R, little y. Nothing else can go with that first one, so go to the second one. Big R, big Y, big R, and little y. It's okay if they repeat. It's totally fine. Sometimes the whole parent will give the exact same thing, so it's okay if they repeat. All right, now how do I fill out my actual Punnett square? I'm going to go through and I'm going to fill out the R's first. So this kid gets big R, big R. This kid gets big R, big R, big R, big R, big R, big R. And then I'm going to go back and I'm going to fill out the Y's. It has a big Y with a little. This one has 2 littles. Big Y, little, and then little, little. Go to the next row, and I'm actually going to skip down a row. So I want you to practice filling out the next one, and I'm going to fill out the one right below it. So big R, little, big R, little, big R, little, big R, little. Then we go back and fill out my Y's. Big Y, little, little, little. Okay, so that'll give you the hang of it. Go ahead and fill out these last 2 rows that I haven't filled out, and I'm going to come back and we're going to solve the problem together. So pause your video, go ahead and practice filling out the rest of this Punnett square, and then we're going to talk about how to solve it. I'm going to go ahead and actually solve it real quick. It's going to look super messy. You can fast forward through this. I totally don't mind. Oop, I made a tiny error. Those are too little. So it gets a little messy, and that's okay. Okay, so now I'm going to go ahead and solve the question. It says round, and it says yellow. So remember what we wrote on the first slide. To be round is a capital R. I don't care what the second R is. To be yellow is a capital Y. I don't care. So how many had capital Y and capital yellow? So if I notice all of these children going down this column right here have capital R's and capital Y's. The kids going down this column also have all capital R and capital Y. So that means 4 and 4, so 8 out of the 16 children will be round and yellow. Box this in, this is your final answer. I'm going to show you a quick method to do this a little bit easier and a little bit faster. So we're going to do one Punnett square for trait 1. So this will be Punnett square for round and square. This one will be for trait 2, so it'll be for yellow and green. So parent 1 gave me a capital R with a lowercase. Parent 2 gave me 2 capital R's. We're doing this just like we've done our other Punnett squares. Go ahead and fill in your Punnett square like normal. And then we're going to move to the Y's. Parent 1 gave me lowercase y, lowercase y. Now notice, if I write parent 1 on this side, I have to make sure I write it on the side for both of them. Can't mix up which side your parents are on. That will get really confusing. Parent 2 is over here, so capital Y with little y. Fill in your Punnett square. Again, make sure that your letters do look different. So I've kind of made my Y's look pretty similar over here on the side, and so it got a little confusing for a quick second. So please make sure your letters do look different. All right, and then let's go ahead and again solve our problem. So we're looking for round and yellow. So again, with all the capital R, don't care what the second R is, and then capital Y, don't care what the second Y is. So in my first box, 4 out of 4 have capital R's. Times, if I look in the second one, how many have capital Y's? 2. So I say 2 out of 4. And again, we're talking about the kids right here. So it's these kids, and then we're talking about all of the kids here. Okay? All right, so let's go back to our math. So when we do this multiplication, I do 4 times 2, which equals 8. And then 4 times 4, which equals 16. Same answer, shorter Punnett square method. Whichever one feels more comfortable for you, that is fine, but please make sure that you do know how to actually read the big Punnett square because that will likely be on your STAR exam.